Welcome to our lunch presentation. The speakers are Athan Kuliopulos and Lidija, who are PIs at the Molecular Oncology Research Institute at Tufts Medical Center. I've been working with both of them for, I guess, about 8 or 10 years. They're very inventive. They were co-founders of Anchor Therapeutics and invented at Tufts Medical Center the pepducin drug development technology, which was the founding technology for Anchor Therapeutics. And today, though, they'll be talking on the topic of R01 to IND. So they are both have a truly innovative story on taking a pepducin compound, PZ128, with federal funding, all the way to an IND and beyond. And you'll hear that story from both of them. So, please.
Thank you. So first of all, I would like to thank the organizers. This is such an incredible workshop. And we're really very delighted to be part of this. So today, Athan and myself will give you a little bit of the background of how we started this entire project, and then from the idea to development of the technology, the introduction to the type of disease and the disease models that we are interested in, and the high risk and unmet need that we recognize, and our own interest in the labs, which is research on platelet and thrombosis. And then Athan will tell you about all the work that was done towards the IND and the whole process, towards the IND and beyond. Can you hear me? Okay. Maybe I should sit down, huh? So just to begin with, so what we are interested in, the high risk unmet need is in thrombosis, and we are interested in acute myocardial infarction. So this is an example of just a schematic of acute. Let me just see if this works. If you have acute myocardial infarction, it really is in lay terms called the heart attack. It is an interruption of oxygen supply to the heart, as you can see here, which results and causes death of the heart muscle. And this is actually the leading cause of death in men, much higher than cancer. So as you can see here on this graph, this is just looking by age, there is much higher prevalence, or much higher number of cases of death per thousand in this particular due to heart attacks rather than to the cancer. As you become older, as population becomes older, it's really a prevalent disease, and there is an increase of bleeding risk, and this is very important because the drugs that you want to develop should have minimal bleeding or should have no bleeding at all. And this is maybe not a good slide to show you during lunchtime, but so this is the cross-section showing you what the thrombus looks like in ACS. So this is looking at the ST segment elevation or non-STS segment elevation. The message I want to show you is that you get this complete reocclusion of the vessel by thrombus. They just completely, this is more than 90% occluded, where also, if you look at this thrombus, you have part of unobstructed lumen, and the thrombus is right here. So these kind of patients need to undergo PCI, or percutaneous coronary intervention, where you can actually insert catheter through the femoral artery, and then you guide it towards the heart, and you can do angioplasty and place a stent, and basically to relieve this kind of occlusion. And oh, sorry. Okay. I hope you can hear me better, or no? Oops. I don't think I have a belt. There. Okay, so is this better? Okay, great. So this is just looking at the number of procedures. And so really, if you look at over time, there has been a great increase in the number of procedures and interventions. So that if you, for example, just look at the PCI is marked with an X here. And especially in the last decade, it's been dramatically increased. So the mechanism of thrombus formation can be simplified on this graph. We can divide this into thrombus initiation step, where you have this primary platelet activation once when this is the blood vessel. If you have endothelium that's denuded, you'll have collagen exposed underneath of this, and this is the primary agonist then for the primary platelet activation. So that's an initiation step. You have to really make the distinction between those steps because the novel mechanism that was discovered in Athan's laboratory is really the second step, the thrombus growth step, which is really platelet-platelet interaction. So first is platelet subendothelial matrix interaction, and the second step is this platelet-platelet growth interaction. For the platelet-platelet growth interaction, there are these secondary platelet mediators. First of all, you'll have thrombin generation initiated through tissue factor, which I'll come back to later. But the secondary platelet mediators are ADP and thromboxane. And so we have, if you look at the platelet, these are the major receptors shown on the surface of the platelet. So for example, the thromboxane receptor, just schematically drawn here, that's the target of aspirin. So the production of thromboxane is blocked, and this everybody, so many people take aspirin. And then the second major drug that's been widely used as an oral drug is Plavix, that hits P2Y12 receptor, and there are other derivatives. The other drugs have been successfully developed against that receptor. And all of these receptors feed through the integrin to B3A integrin. That's your final step. There are many inhibitors against 2B3 integrin, but in this case, you will end up with very severe bleeding. But in high-risk patients, you have to really give them an extremely high regimen of protection to protect against activated platelets. So the receptors that we are interested in are thrombin receptors. This is protease activity receptor 1 and protease activity receptor 4. They are both protease activated, as I mentioned. The difference is in affinity. FAR1 is a high affinity receptor, whereas FAR4 is a low affinity receptor, so they actually have very distinct functions. And if you want to develop a drug, the FAR1, the high affinity receptor, is the one receptor that we were interested in, and not the low affinity thrombin receptor, because based on a lot of work that we've done in our laboratories, FAR4 is a safety mechanism. You really don't want to cross-block the FAR4 receptor. And so looking schematically, the question is, can one develop an inhibitor against FAR1 that will prevent platelet activation and arterial thrombosis? So this is just schematically showing a vessel. Again, like I mentioned before, what's important to know is that if you have an injury, an assault, or during atherosclerosis, if you have an atherosclerotic plaque, a vulnerable plaque, and if this plaque is dislodged, you will end up with exposed collagen, which will then result in tissue factor-mediated thrombin production and generation of thrombus formation. And as I mentioned, there are those initiation and propagation mechanisms. However, what was really important was not only that there is thrombin, the discovery that was made is that platelets actually can be activated by completely another class of proteases, which was not appreciated before. And those are matrix metalloprotease 1 is the matrix metalloprotease that activates also FAR1. And it cleaves it, and I'll come back to details of this. So this is, the red shows you fibers of collagen that were just placed on a surface of a glass surface. And platelets were labeled in green. And then using the arterial shear stress, you basically just do the flow and look at the adhesion of platelets. And as you can see, these green spots, those are actually those propagating platelet aggregates that I mentioned on the schematic. What was really pretty incredible and novel here is that if you use collagen, first of all, also we found that platelets contained already pre-bound pro-MMP1. The MMP1 has to be activated through a cascade into an active form. And so the platelets, this is just showing you fluorescently labeled MMP1 on the surface of a platelet. This is with zero collagen, the basal level. If you then incubate this with collagen, you displace this pro-MMP1, and it actually is activated into active MMP1. And I'm not going to go through the details of the paper, which actually showed that this MMP1 activates PAR1. But what I wanted to show you, what was extremely exciting, is that actually MMP1 cleaves PAR1 at the extracellular domain, but at a different site, the thrombin. And the way that we could show that is if we synthesized a small TR26. This is just a small peptide that has a thrombin by a cleavage site and also some of the adjacent regions. And you can see the thrombin releases the typical peptide, but the MMP1, it came as a surprise, did not cleave at the same site. And this, we just also made some of the mutants to show, identify the site. This is modeled from MMP1 by Andrew Bohm, and this is a complex between a catalytic site of MMP1 and our PAR1. And what's really beautiful is that you could find this cleft region in the active site. So this TLDPRS, this is the cleavage site, and then PR peptide is the difference between thrombin. Thrombin cleavage site is at the SFLLRN, is the ligand that you can also make as a peptide. And as a matter of fact, we also made this peptide, PR SFLLRN, and we could study different properties as a surrogate for the two different proteases. So what we want to do is to develop an inhibitor against PAR1, but that can inhibit both MMP1 and thrombin platelet activation, as shown here. And so that presented to us an opportunity. We have an unmet medical need, despite I mentioned that there are really good inhibitors against thromboxane and ADP receptors. So now what we have done is we have identified another whole class, a third receptor that's an endocrine response, MMP1-driven PAR1. So it seemed like this would be an incredibly good target because despite aggressive antiplatelet and anti-therapeutic therapies, the very procedural adverse effects in clinic. So during the procedure, you can also, you can imagine you have to protect the platelets from activating, even just at the procedural level. So there were still fairly high clinical adverse effects, that consist of death, MI, and stroke. So there is a lot of room to develop new therapeutics for the patients that are undergoing PCI. And so the novel agents are needed for the reduced clinical events without adding a bleeding liability. As I mentioned initially, especially as the patient's population ages, the bleeding liabilities become higher. So you don't want to block, there are also thrombin blockers, but you want to block with alternative pathways rather than thrombin, because thrombin would result in severe bleeding. And there are no agents that target the collagen-initiated pathway that are in clinical stage, and so we have identified this as a collagen-initiated pathway. So the overall goal of this project, so what we wanted to do, we knew that we could go with the intravenous administration because the PCI of the nature of the entire procedure. So we thought we could develop an intravenous antiplatelet agent for ACS patients that are undergoing PCI. So we knew our patient population. We also know that we want dual mode of action. We want something that has really fast onset, because when you start the procedure, you want your drug to work on immediately and to rapidly protect platelets. And our target is a platelet, which is really nice because platelets you can study very easily. So you don't need a biomarker. You have a direct cell, a target that you can actually look at the activity. So your PK and your PD you can easily measure and design. Well, designing is harder. And you also want a reversible mechanism of action because of the bleeding risk. So all these procedures are not only using one drug. You use multiple drugs, and depending on the risk of the patient, the clinicians decide what kind of combinations to give and how protected the platelets should be. But if our drug is reversible during the time of the procedure, so to protect for 1 to 2 optimal hours, that was what we were aiming for. And then if we had a good drug and a method to make it, then it was to find money, to do all of this, and conduct IND-enabling efficacy, safety, toxicity studies. Required thing is to go into 2 species. And Athan will go in detail on this, because in our case, our receptor is not present in rodents like mice and rats. It's only on the platelets. It's expressed on other tissues. So we had to actually conduct the species that we had to use was monkey, and that becomes very expensive, as you can imagine. But now we are going through NIH-funded work. And the second species had to be guinea pig because they also have PAR1 on their platelets. And then following IND, you have to know exactly what your patient population is and what is going to be your design of phase 1 and phase 2 trial. Because to conduct an IND, you have to be able to cover both phase 1 and to go into patients. At the end of IND, you are ready to go into your phase 1, which should prove that you can, and then go into the phase 2 immediately after that. So the big question is, you've heard this morning, the big pharma, they have enough money to do this, and they have to convince, I guess, internally that it's worth doing. CTI brings you up to a certain point. And we try for every opportunity as well, depending on the question. But can this be done all the way from designing a molecule to an IND and even beyond, going to phase 2? Can that all be done through NIH? And so the technology that we've been working on is now about a decade old, 10 years. And so this is just a schematic of our GPCR, protease active receptor 1. We like to let it just for once float in the space because it's so beautiful, but it was modeled on rhodopsin. The beauty is that more and more crystal structures are coming up, and we were lucky that PAR1 is a really good candidate to model it on rhodopsin. And it's a 7 transmembrane receptor. And Athan will discuss, this is our region of interest. This is our molecule that's modeled onto PAR1, and it's based on the intracellular region. So our technology is going to target intracellular region of the receptor. And so G protein coupled receptors. So PAR1 is a G protein coupled receptor. These are the major drug targets, if you look. And so this is one of the most important classes of signaling transduction receptors, and about 30% of all approved drugs target GPCRs. And this generates really a great amount of money and sales. One is compelled to say this, but I was really amazed with the talk this morning on the cystic fibrosis, because it wasn't presented that this had to make $1 billion. So to make a good drug, I mean, you just want to make a really best drug that you can for the patient population that you know could benefit. And so there is huge untapped potential, but in broad prospect, this technology could be used for any GPCR. And only about 30 of 700 known GPCRs have actually been successfully targeted. And the challenge is that it is really hard to identify small molecules that will disrupt these large ligand interactions. There are also tethered ligands, for example. Like in our case, protease-activated receptors will have a generated tethered ligand, and that's completely different. So, for example, there were screening strategies using peptides instead of tethered receptor that would identify really potent molecules, but when you went into actual in vivo studies, it wasn't very efficient against the natural ligands, which are proteases. And you want to obtain desired specificity, so our approach is peptide-peptide interactions. So it is very possible to build in specificity. And in case of receptor dimers, heterodimers, and oligomers, this makes it very complicated in screening small molecules. Also, we can actually identify agonists, not just antagonists, and it can be used for deorphanizing receptors. So this is just showing you, these are all the GPCRs, and we are focusing on the rhodopsin family of GPCRs. And the ones that are highlighted, the crystal structures are known of these. And we wish that our PAR1 would be known too, but so far it's been a higher challenge to get a crystal structure on this. And so this is just now showing you schematically to simplify this. The GPCRs have 7 transmembrane domains, the 4 extracellular loops, the 3 intracellular loops, and the 4th one is really special in some cases where it has another 8 helix. And they interact with G proteins, which consist of G alpha, beta, and gamma subunit, shown here. Although this is schematically, this is really, we just put it on the side. Usually you have a heterodimer to GPCRs, and G proteins are so large that they can actually, you can position 2 GPCRs on top of the G alpha, beta, and gamma subunit. And with G alpha, you have different types of G alpha 12, 13, Q, I, and S. And this is just showing you some of the attempts or some of the successful interactions that we could inhibit in these receptors that are marked with red that we tested using our technology. So small molecule, again, to come, you can target GPCRs to small molecules and monoclonal antibodies. And pepducins, so they are specific because the way they differ is we actually thought we want to target this intracellular region as a new way of targeting signal transduction. So to do that, you have to imagine that you have to be able to transfer across the membrane and then interact with the receptor. So to do that, we have conjugated a lipid moiety. So the pepducin really consists of a synthetic peptide, which is usually 7 to 15 amino acid fragment of GPCR, of the intracellular loop. It could be any of the loops, and which nature engineered already specificity in it. And then this hydrophobic moiety, which can be palmitate or any other lipid. And this has already, actually someone has measured this is an extremely fast event, this flipping across the membrane. It's in milliseconds, and it then inhibits G protein coupling, or depending on the design, it can be an agonist, depending on the receptor you want to design this against. And so this is, I think, just showing the same thing. And what was very fascinating is when from X-ray crystal structures is that because palmitate is naturally occurring in our body, and it's a component of a membrane, so that they're actually binding, surface binding sites within the receptor for palmitate. So which indicates that it's not only the peptide that we can modulate, but also the lipid it would be possible to modulate both peptide and lipid and vary the properties of the pepducin. So this is a very historic piece of data that dates back probably 10 years ago now. The very first time we designed a whole third loop of the intracellular PAR1, what was fascinating is adding it to the platelets. Now this is looking at calcium, is that it resulted in this sharp peak. So if you stimulate platelet with thrombin or with SFLRN, which is just the ligand, you can make a peptide to activate PAR1. Then when you looked at pepducin, it was possible to actually make an agonist. So in our very first experiment that we've seen, we realized that the pepducin technology can go much further beyond. We were interested in inhibitors, but that it actually can make an agonist as well, depending on the sequence. Oh, I'm going. Okay, yeah. So the reason I told you initially, there are two receptors. There is protease active receptor 1 and protease active receptor 4. They have different affinities. So for other purposes, we were interested in being able to inhibit each of the receptors selectively. And so we made these pepducins. I mean, we made a number of pepducins, but these are the two that we characterized here against PAR1 and against PAR4, which we call P1 PAL12 and P4 PAL15. And if you use, again, calcium is our primary assay. We then go to aggregation and animal models and some other assays. But calcium has proven to be an incredibly effective way of to screen at least protease active receptor pepducins. And so this is showing you the calcium signal for the two receptors, PAR1 and PAR4. And you can selectively inhibit PAR1 or PAR4 in this. And so this is as much as I'm going to tell you. Just an intro of, I think, more like a historic data. And then Athan is going to tell you now what we've done actually very recently with this.
So in the next few slides, I'm going to take you through our latest data, which is not published. In fact, it'll be coming out in press in a couple weeks in circulation. And as Lidija just explained to you, sort of the history of pepducins and the potential to target, in principle, any G protein coupled receptor and even other transmembrane proteins or membrane-associated proteins. And as she's told you, we're going after PAR1 as our, quote, what we consider our low-hanging fruit. And what we did through primarily R01-funded research, we made over 50 different pepducins. And the pepducin that had the right kind of properties that we wanted, a lot of it was obviously efficacy and specificity, but also pharmacokinetics was very important. And Lidija had a paper published last year, which we made several different kinds of pepducins and looked at PK. And this was the pepducin PZ128, which I'm going to focus on, which is a 7-amino acid peptide from the third intercellular loop in part of the juxtamembrane region on the N-terminal portion of transmembrane 5. And down here in the left-hand corner is a high-resolution NMR structure, which is solved by our collaborator, Jim Balea, who's in the audience in the back there. Hey, Jim, thanks for coming. And we had successfully formulated this pepducin in an FDA-approved vehicle. And it's extremely stable at 4 degrees. And actually for several months, it's extremely stable and keeps the same particle size. And this is the C16 palmitate over here, which is actually not structured except for the first couple carbons coming off the attachment point. And here's a 7-amino acid helix. And what we were very surprised to see is, first of all, it was extremely well structured, and it formed a beautiful alpha helix. And in fact, this alpha helical structure superimposed almost perfectly with the analogous region from this intracellular portion of transmembrane 6. And this is the part of the receptor which is going to interact with G proteins very well, with the C-terminal portion of the G-alpha tail. And in the extensive work we've done on modeling homodimeric interactions, we also believe this portion of the receptor interacts with the 8th helix in a homodimeric interaction. And down here is just showing the superposition of the pepducin in green with the C16 palmitate here going into the membrane with the alpha helical structure at the peptide backbone of PAR1. And this is just some of the early efficacy data which we did with isolated human platelets. And I'm showing you here platelet aggregation, which is light transmission aggregometry. And as you add increasing concentrations of the pepducin, you can completely inhibit aggregation. When you get above 1 micromole, it's completely blocked to SFLRN, which is the PAR1 peptide that Lidija mentioned. It does not have any activity at all against three of the other most common GPCRs on platelets. PAR4 is the most functionally homologous receptor. The other thrombin receptor, it has no activity in platelet aggregation on PAR4. And I'll show you it had absolutely no activity on PAR4 in guinea pigs and in primates. It also has no activity on the ADP receptors or the ristocetin receptor. And as Lidija mentioned, if you screen against SFLRN, and several drug companies did use SFLRN, the peptide, as a screening method, you can actually create a beautiful small molecule antagonist against SFLRN. But the real agonist is the tethered ligand, which is generated by thrombin cleavage of the receptor or MMP1 cleavage of the receptor. And this is an example of thrombin curve. So now we're adding increase in concentrations of thrombin. You get full platelet aggregation in squares, which is just vehicle. This is RWJ. This is a small molecule peptidomimetic, which was generated by Johnson & Johnson, our collaborators in New Jersey. And you can see you get about a little less than a twofold shift on the thrombin curve when you add the RWJ compound. And this is the pepducin, and you can see that you actually get slightly better shift to about, you need about 10 times more thrombin concentration to activate the receptor. This is completely blocking PAR1. What you see here at high thrombin concentrations is the second lower affinity thrombin receptor kicking in. So this is what Lidija alluded to. At higher thrombin concentrations, we believe this could potentially be a positive thing, that you would be activating the PAR4 thrombin receptor, which is probably very important for primary hemostasis and reducing bleeding liability. So some of the first experiments we did were in guinea pigs, and the other rodent species that we tested and other people have tested, mice and rats, unfortunately do not have PAR1 on their platelets. They have PAR1 in other cells like endothelium and smooth muscle cells, but they absolutely have no PAR1 response. So the only appropriate small animal species, or actually any animals except for primates, are guinea pigs. And this is just showing that the SFLRN peptide ligand will activate, in this case, up to 80% of guinea pig platelets. And this is PZ128, the palmitoylated 7-mer from PAR1. And what we're doing now is we're infusing in either vehicle alone, 3 or 6 mg per kg pepducin into the guinea pig in a 10-minute intravenous infusion, then withdrawing blood at different time points. And as you can see, a nice dose dependence where you block about 3/4 of the aggregation by 3 mg per kg and about 80 or 90% at 6 mg per kg. Again, specificity was shown here with other agonists. This is a thromboxane mimetic, and this is ADP. Has no inhibition against these two other agonists in guinea pigs. We then went to, that was a pharmacodynamic marker, namely platelet aggregation, and then we went to a disease model, which is arterial thrombosis on carotid arteries in the guinea pig. And what I'm showing you here is actually not a guinea pig, but I think this is from a mouse. But this is the concept that if you have the lumen of a blood vessel, we're staining the endothelium. If you injure them with ferric chloride, it actually burns the endothelium and exposes collagen. So we've injected an antibody against collagen into this mouse, and you can see collagen exposed, and then the platelets are labeled with a fluorescent dye, and you can see accumulation under arterial shear stress. So we're basically going to be doing the same experiment, but in a much larger blood vessel in a carotid artery in a guinea pig, where we're driving this through collagen. And what we're doing here is infusing for 10 minutes, creating the injury, which in this case lasted about 20 minutes with ferric chloride. We remove the ferric chloride, and then we place a Doppler probe distal to the injury, and then we measure time to occlusion, which we can do either up to 60 minutes, which is a predefined endpoint here, or up to 90 minutes, which we did later when we were looking at synergy with clopidogrel. And what you can see in an untreated or vehicle-treated animal, the time to occlusion is quite rapid. So here's an animal whose carotid artery has been injured. These animals actually occluded even during the injury point, and all the animals were occluded before 20 minutes, complete occlusion of their carotid artery. If you infuse 0.05 mg per kg beforehand, you get no effect, but even when you go to 0.075, you can get a statistically significant effect, at least in the median in these animals, and that effect is persists at higher concentrations. So we know that we can infuse in the pepducin and protect against arterial thrombosis in the guinea pig. Since in our upcoming clinical trials, standard of care treatment is Plavix, at least one of the drugs, also aspirin, and Plavix, otherwise known as clopidogrel, blocks the P2Y12 ADP receptor. And what we did here is we looked for any synergistic effects with Plavix, so hitting two pathways, hitting the P2Y12 ADP, which activates GI signaling, and the PAR1 pathway, which hits 12-13 and GQ signaling pathways. And what you can see here is at low dose pepducin, this dose here, there's no effect. This is a low dose clopidogrel. You get a slight increase, but it wasn't significant with 1 mg per kg pre-treatment with oral clopidogrel. However, when you add the subtherapeutic dose of pepducin plus clopidogrel, you see this great increase in prevention of arterial thrombosis. So this is sort of our one of the endpoints, obviously, we like to meet when we go into our phase 2 studies with patients that are taking clopidogrel. So the second species that we looked at for efficacy was baboons, and these studies were done over in Oregon with Andres Gruber. And Andres and his colleagues have a really phenomenal track record of testing drugs, pretty much all the drugs that have been approved in the last 10 years in their baboon models. And what we did was a pharmacokinetic study where we had, in this case, I guess we had 5 different doses from 0.2 up to 6 mg per kg. And these are just single time points at 1 hour, but we did several other time points with 3 mg per kg and 6 mg per kg. And we did a either 30-minute infusion or 15 minutes on these low doses, or 45-minute infusion on the higher doses. And what you can see is a rapid increase in plasma levels. These are in thousands of nanograms, and it's about 1,000 Dalton molecular weight. So this is equal to about roughly 4 micromolar. This is 8 micromolar, 12 micromolar, etc. So the property of these pepducins with a lipid tag is do not have a property of a small peptide. They have a property more like a lipid. They have beautiful pharmacokinetic properties, rapid increase and sustained levels as long as you have the infusion. And eventually you can get a terminal elimination rate. In the case of the baboon, it was between 50 to 80 minutes. We've also done this. These are in 10 to 12 kilogram animals. We've also done this in smaller cyno monkeys, and the PK is almost exactly the same in the 4 kg cyno monkeys. So it looks like we've got really optimal properties, rapid onset of action, high sustained levels during the infusion. The PCI procedure averages usually less than 1 hour. You turn off the infusion, and then you get elimination, a half-life about, let's say, 1 to 2 hours roughly. And the other key point is here. The patient is going to come out of the cath lab, go to the floor for observation, usually overnight, and discharge the next day as a typical course. And there really was little to no pepducin detected after 8 hours, which in plasma, that is. And this is actually a good property because now the patient we're not blocking their PAR-1. If there's any risk for bleeding, this is where the risk for bleeding can come in, and the patients are usually treated subsequently with oral clopidogrel or prasugrel potentially, and aspirin, thereafter because they have a stent implanted. And this is showing the PD biomarker. So again, as was mentioned in the earlier talks, one of the key factors to seeing whether or not you have efficacy in your patient population is a reliable pharmacodynamic marker. In this case, we're looking directly at the target receptor on the blood platelets at different time points. So it's really a very nice method to see whether or not you've hit your target dose. And so what we're looking here is baboons that were given these 3 doses, 1, 3, or 6 mg per kg over 45 minutes, and withdrawing blood from the baboons 0, 1, 2, or the next day, 24 hours, and looking at inhibition to the PAR-1 agonist here. And what you can see is at 1 mg per kg was subtherapeutic. There really wasn't any appreciable inhibition even at 1 hour. However, when you jump up to 3 mg per kg or 6 mg per kg, and this is at the lower dose of SFLRN, you can see a dose dependence here of blocking aggregation. And at 3 mg per kg, we believe is roughly the therapeutic dose. You can achieve about 90% of inhibition to PAR-1 at the 1 and 2 hour points. So here you can see the drug was stopped at 45 minutes, and at 2 hours you still have enough drug on board, especially in the platelets, that can prevent the platelets from being activated by thrombin. This is an ex vivo experiment. And again, by the next day, 24 hours, there really isn't much platelet inhibition. This is looking at specificity to the 3 different agonists here. Here's PAR-1 again, pretty much the same data here being blocked. However, the PAR-4 receptor, ADP receptors are not blocked at any time point. And so that's nice. We can do baboons. What about platelet aggregation? What about arterial thrombosis? So what Andres has done a beautiful job of is developing a high arterial shear stress model, which very closely mimics the flow characteristics of a human coronary artery. And he has 2 different size stents that he uses, or rather lumens he uses a 2 millimeter or 4 millimeter diameter shunt, and he labels these baboons with radioactive platelets and radioactive fibrinogen, and you can independently, using 2 different detection methods, count the platelets and the fibrinogen. And what you can see here, at least the people in the front of the room, is flow is this way. You can see here implanted is a little Dacron graft, and the platelets adhere to that. And then you see in red here are the platelet-rich thrombi, which actually can get up to 8 or 10 centimeters long. This actually happens in humans also. So you have the initial area where the platelets are adhering, and then the platelets will grow, and then this can embolize. So you can see actually platelets forming and then falling off at different time points. So what we're doing here is, again, we're doing infusion of 45 minutes. And here we're comparing on this top panel here, vehicle alone, we're looking at accumulation of platelets over a 1 hour time period. And if you put in 3 mg per kg, you get about 30 to 40% inhibition, which was not quite statistically significant. However, at 6 mg per kg, we got p-value less than 0.001 at 6 mg per kg with about 40% inhibition at the top part versus vehicle. So this is actually very typical of the other PAR-1 inhibitors that have been tested. They generally, in this model, give anywhere from 10 to 40% inhibition. And then, I think this is my last data slide, the other critical point is we do not want to affect hemostasis, namely bleeding. So in these animals here, we're looking at, these are all male baboons, 3 different doses, and we did several measurements. I'm just showing you platelet count, hematocrit, and bleeding time. If you, there was no statistical differences between baseline and platelets drawn 1 to 2 hours after the infusion. So we didn't lose platelets, or we didn't change the hematocrit, and most importantly, we had no effects on bleeding time in any of these animals. Interestingly, if aspirin and clopidogrel were run through this as a safety, they would probably both be rejected today. These cause massive bleeding in baboons and actually other animals too. So humans don't quite bleed as much as primates, but primates are very sensitive to thromboxane and P2Y12 inhibition. Oh, sorry, I have one more slide here. So the last slide is in collaboration with Dr. Carrie Kimmelsteele, who is Chief of Interventional Cardiology here at Tufts Medical Center. We did a clinical study looking at bivalirudin and its effects during PCI, which was actually a very valuable study for us to design a lot of our work with the pepducin, because it's a peptide, it's a 20 amino acid peptide. We measured actually, we were the first ones to measure the pharmacokinetics and pharmacodynamics of bival using the approved dose in ACS patients undergoing PCI. And what we measured in bival is that this is a nanomolar inhibitor. However, it does not work until you get into the micromolar range, and that was the big finding for bival, that it had to be given as a bolus, then an infusion in order to achieve therapeutic levels, which were between 2 to 5 micromolar in blood. However, bival is a direct thrombin inhibitor. It's designed to be a direct thrombin inhibitor, and what it does is when you give it to a patient at appropriate concentrations, you actually elevate the activated clotting time. So this is a desired effect, that you put a direct thrombin inhibitor, which is great. You protect against thrombin inhibition. You turn off the bival infusion. Eventually, you lose, over time, activating clotting time. However, the pepducin does not prevent thrombin. This is a verification of that. In patient blood, even up to 150 micromolar, you have no prolongation of the clotting time. Okay, so now I'd like to, the last couple slides, this is sort of the title of the talk, how did we get here? What was the procedure to get to the point where we're ready to enter clinical trials? And as Lidija mentioned, it is possible to fund an entire project in your own lab here at Tufts through the NIH. And basically, what you're looking at here was the timeline. We're right about here, actually, on 30 months out, about two and a half years out from starting the RC2 study, funded study. And basically, through entirely NIH money, we're able to synthesize, screen pepducins in a few months, get a selective inhibition of platelet PAR1, and then we were able to find a vendor that was extremely talented and was able to do about a 50 to 70 gram synthesis of PZ128 at extremely high quality, greater than 99% purity. And we initiated stability studies at 3 different temperatures, including room temperature, and thus far, 2 years later, almost 2 years later, it's still 99% pure and stable and active. So again, even GMP material, unless you're a very complicated small molecule, is certainly doable within a budget of what the NIH can give you. We then were able to conduct guinea pig and efficacy studies, initially with non-GMP material, but then literally within a month, we already were doing, all of our studies were done with the GMP material. And we started 7-day repeat dose, high dose monkey tox and safety studies a year out with GMP material. We did a lot of other studies I'm not talking about. We went to the FDA in February of last year and had a pre-IND meeting at the cardiovascular portion of the FDA, which went extremely well. They asked for, of course, additional studies, which we had to acquire some more money from the NIH. And so we went to the SMART program, which I recommend you check that out if you're involved in the Heart, Lung, Blood Institute kind of research. And they just funded, early part of this year, the rest of the remaining studies that the FDA asked for, which included pivotal studies in guinea pig under GLP conditions, which we just finished, which have worked very well, and doing a few other studies. So we hope that by the end of the summer, we'll be submitting our IND. And I would also point out that we didn't do this by ourselves. We had a very large team of people, including regulatory experts, and we can talk about that at the end of the talk if you'd like. So it's very important that you assemble other people that are very familiar with the FDA if you don't have the luxury of being at Pfizer or Vertex or Sanofi. And now for the second part of the program, so now once you get through all the preclinical work, the efficacy studies, the safety studies, file your IND, of course the most important part of the clinical development program, and we're lucky to receive a grant from the NHLBI to fund these studies to do phase 1 studies in about 35 to 40 patients, and this is scheduled for next year, and this will be a dose escalating study at several different doses run by our Data Safety Monitoring Board, which will be external. This will be done actually in Baltimore, and looking at safety in the patients, tolerability of the drug, looking at the pharmacokinetics, looking at the pharmacodynamics exactly as we just did in our other primates, and looking at antiplatelet efficacy, so we can choose our doses and IV administration for our large phase 2 study. We will have to make more drug at that point. We'll be making it even here. And right now we're planned to do 800 PCI patients. This will be a multi-center study. We have at least 3 hospitals that will be doing this, and we'll be looking at primarily safety, bleeding especially, with the experiences of Vorapaxar, looking at a subset of patients, looking at aggregation, and about 1 to 200 patients at least at different time points, different biomarkers including MMP levels, TAT levels, thrombin levels, inflammatory markers. And then, of course, standard efficacy endpoints will be 30-day endpoints of major adverse coronary events, including myocardial infarction, death, urgent revascularization, and stroke. And this will go on for a few years, and eventually we hope to be successful and go on there to phase 3 studies. So in conclusion, Lidija's talked quite a bit about the technology in general and how we got to where we were. So I hope we've given you a taste of how we can target the intracellular receptor G-protein interface with lipidated peptides. In principle, it could be applied to any GPCR, and it has been successful in over 20 different GPCRs now. The one that we've been working on, PZ-128, has favorable pharmacokinetic and pharmacodynamic properties in primates and guinea pigs as an antiplatelet agent. It does not have any bleeding liabilities that we can detect or abnormalities on coagulation in baboons, and has been well tolerated in monkeys. And last but not least, and probably the most important, is that yes, the NIH can fund this early translational research program in your own research group if you apply several times. And just like to stop and acknowledge the large number of people that did this. This is definitely a team approach, not quite as large as Vertex, I don't think, but with all those different people. But I'd like to, first of all, thank everybody in my lab and Lidija's lab, our key collaborator, Jim Balea, who did the NMR structure. Ping is the senior scientist in our lab who did all of the guinea pig work, human platelet work, the whole bivalent Ruden study. I mean, she's really quite amazing and has been absolutely pivotal. Shogo has done a lot of the safety studies with Ping, and we work pretty much on a I won't say a daily basis, but a weekly basis with the CROs that are doing the safety tox and pharmacology studies at the CROs. And Vishal, I would like to mention, was a postdoc in my lab who's now got his own lab back in India, very, very brilliant scientist who was the first author on the 2009 Cell paper, the discovery of the MMP1 PAR1 connection, and that was really obviously an important contribution for getting people excited about this project. Kerry Kimmelstein, I don't think he's here, has been a long-standing collaborator. Lidija and I have done several studies with Kerry. He's director of adult interventional cardiology and is the major PI on the, he'll be the major PI also on the phase 2 work being done here at Tufts. And also mentioned we, in the past, we've collaborated with even Vertex and other companies looking at anti-platelet inhibitors, medicines company. And, Also not on here is Andrew, who's right here, who is our X-ray crystallographer collaborator. And on the Oregon side, here's Andres, who does the baboons, and his very talented primatologist, Ula Marzek. Merrill and Pat were the two key people to really guide us through the regulatory part, all the things we had to do, hooking up with the right CROs. So these people, I could give you their contact if you're interested to do the IND work. And there's many other people also we've worked with. The Baltimore team, this is Paul Goebel. And if you guys know Paul, he's really an incredible guy, director of cardiovascular research at Sinai Hospital in Baltimore. And it was his team, especially Kevin Blyden, who helped us put together these very complicated clinical trial protocols. Some of them are, approaching 100 pages long, looking at different kinds of bleeding that can happen. And Paul and Kevin and their team have been very important. So again, I think as I said several times, you need to have a large multidisciplinary team approach to be successful. Of course, I'd like to thank John and Nina for all the great work they've done in the past and continue to do, and for inviting us and throwing this great workshop here today. And of course, funding wouldn't be here if it wasn't for R01, RC2 programs, P50, SMART programs, etc. And again, I urge you, this is really a big opportunity now. Francis Collins and other institute directors at the NIH are very in favor of translational research, and there are a number of things like the SMART program for each of the different institutes that you can tap into, and you can be very successful there. And I will stop there and take questions. Arthur.
Right. So, mechanistically, targets these proteins to the lipid back. What's known about PAR1 and lipid bags?
So if you can hear, Dr.
So this is a question about palmitate generally targets proteins to the lipid rafts. I don't know much about lipid rafts in platelets, but PAR1 may be, it says a G protein coupled receptor, so it's probably lodged into some kind of a detergent-resistant raft structure on the membrane. Is it possible that pepducin is going in and dislodging this PAR1 from the lipid rafts? So if you, can you use something like a generic lipid raft disruptor to see whether that has the same effect on PAR1 signaling?
So great question. Lipid rafts are always hovering out there like the dark matter of the universe. So lipid rafts, a lot of people work on them. I would say we have worked with lipid rafts. Palmitate per se is not a lipid raft lipid. So in fact, it's actually decreased in concentration. So lipid rafts are enriched in cholesterol and sphingolipids, and we have made pepducins with cholesterol and sphingolipids. So, and we're working actually on a very nice project for special pepducins that can home to lipid rafts. PAR1 seems to distribute in lipid rafts and non-lipid rafts without any preference. In other words, you see it in the lipid raft fractions and the non-lipid raft fractions. Those are low-resolution fractionation experiments using sucrose gradients. If you do a functional assay, for instance, what's it called, cyclobeta-dextran extraction of cholesterol to deplete lipid rafts, you damage usually cell function like aggregation, so it's hard to say whether or not that was because you sort of did your mechanism. So I would say that it's still an open question. I think there's room for pepducins to be tailored for lipid rafts or not. Thus far, we're using, quote, non-lipid raft pepducins like the palmitate. But we also do a lot of work with lipocholic acid, which can have incredible properties. This is a rigid steroid, which is enriched in lipid rafts. So we don't think this is a lipid raft story at this point.
Why so many patients for a phase 2 trial?
Yeah, great question. Yeah, so very different. We probably have a lot of patients here than you're used to. So this is the size of a typical trial for phase 2 in PCI patients, about 800 patients. Yeah, they can be as low as 600 up to 1,000. That's a typical range. The reason why is the endpoints are in the 5 to 10% range. So for safety, you have to get a certain power. Can you detect bleeding? They're really looking for bleeding in phase 2 is the major indication, and a dose dependence. So typically 3 doses. That's what our studies plan, 3 different doses versus placebo. So about 200 patients in each arm, and with a, let's say, 5 to 10% event rate, and bleeding can be from low to high. So that's basically, it's powered to detect bleeding. It will not be powered to detect efficacy. That's the other question is, how many patients in a phase 3 study? It's about 12 to 20,000. Very expensive, a couple $100 million. But well worth it if it's successful. You're talking 1 to 2 million patients easily in the United States alone a year.
So,
Brent?
So Athan, why did you decide to go the NIH route rather than partnering with biotech or pharma?
Well, we didn't decide to do that route. No, we were interested in PAR 1. I guess when Lidija discovered PAR 4, which was in 1998, we thought PAR 1 was going to be a very hot target, and many large pharma companies, including Merck, Portola, Millennium, I guess Schering-Plough. Yeah, we met every single person you can think of, every single large pharmaceutical company and biotech that was interested in PAR 1. So yes, there was a lot of interest in that. At the end of the day, most companies had their own small molecule programs, so we were competing against small molecules that the companies obviously own their own IP. It's something that was a choice which we did not quite. We couldn't bump out their own internal people. So that is, that's an issue which we could talk more about, but we have a lot of experience from the other side as customers, if you will. But I would say we decided to do it because we couldn't get enough funding. I think a lot of it is, if you're looking at phase 2 and phase 3 studies of that magnitude, the risk is even higher. So now you're making, you're looking at about close to $10 to $15 million worth of funding right there. Okay, that's a sizable amount of money, and then phase 3 is much larger. So I think maybe those are sort of the two answers.
So in the end, you'll probably, I assume you're going to partner with pharma on phase 3, or would that be with NIH?
I would assume so. Yes.
I have
Perfect.
I have a related question to Brent. Since you mentioned it's going to cost about $200 million or so for phase 3, and you also mentioned that pepducin technology was about 10 years old, and your phase 3 period is going to be at the end of 8 or 9 years, that's about 19 years. How do you deal, so you're going to need commercial viability. And so since presumably your patents, et cetera, were submitted, when the pepducin technology was initially created, how are you going to generate that $200 million without having a obvious ability to protect your IP at the end of 19 years?
IP is probably not an issue. We have many other patents which will protect the use of the compound, formulations, things like that. Very critical. So we're actually in very good shape. If you're just talking about the general IP, we still got another 10 years. So it's still a long time. But you're right. You don't go into this thinking it's going to take you 19 years, which would be 9 years from now. But that's, I think some large programs. I don't know if there are many programs that were less than 10 years. So I think most people, when they think about it, they're thinking about pieces of it. very few people were in it the entire window. It sort of started the day they started working on it, right? So if they came in on it in 2012, let's say they picked up PZ128, the other stuff was forgotten about. Plavix is a good story because Sanofi, which made a ton of money on Plavix, up to $8 billion a year for a few years, that drug was sitting around in the 90s. That was a drug that was screened and was found as a calcium channel inhibitor, which was erroneous and incorrect. And patents were submitted, and the patent protection just ran out, I guess, around this year. So, so in other words, there are obviously ways to extend patent life, and many compounds do take, 10 years would be, I would say, a minimum, and probably I could defer to our colleagues here from Big Pharma, but I would say probably 15 years might be an average.
Any other questions?
Athan, does it affect GP1B?
GP1B. The pepducin, no.
Yeah.
No. Okay, thank you.
Just a general question. And that is, first of all, it's a fantastic story, and it's wonderful to see it working out as well as it has. So I know you work on both sides and have done a fantastic job with both. But given where NIH is, I mean, just your opinion, do you want to see them supporting clinical trials? I mean, can we afford to have them supporting clinical trials? And if you look at the success rate of developing drugs, once it gets to the stage you're talking about, my understanding is it's like 1 in 100. So can NIH really afford to be doing that? And that's not just a question for you, but a more general question. Of course, I'd like to hear what you have to say.
Yeah, that's a great question. Obviously, clinical trials of this size, phase 2 is within reason. Phase 3, probably not for this size. I think that's it depends how much it costs. So I would say the opposite. I would say we can't afford not to have the NIH fund clinical trials because of all the problems that we talked about. I think that is happening. NIH has always funded clinical trials. And I did look recently, a few months ago, on the percentages, and I was actually pretty surprised. It's a sizable amount of funding goes to clinical trials in the NIH. I don't know the exact number, but hundreds of millions of dollars at least just for NHLVI alone. So there's a very large component already there. And I would say this is something that's new in a way, that you're looking at programs where the NIH has identified this huge problem, and they're putting money into it. So the NIH developed this translational new institute. There's actually a new institute where they have created from other institutes, and it's going to have its own structure, which is to do this kind of work. So bridging from drug discovery into phase 1, phase 2 studies. So I think the NIH will take a part of their, that'll be part of their research portfolio, and I think it's larger. But they're not stupid in putting it all into that. So I think it's a mix, it's a balanced portfolio, but it was unbalanced. It was very little of that. And this kind of stuff was fairly rare. And that, I would say the opposite. I would say the problem is if you have a really good idea, and I know you do, the basic science study sections are not going to like it. They'll say, oh, yeah, you can easily go to a pharmaceutical company and do this. So I think that's why you have to have special funding mechanisms besides SBIR, which is, again, very different kind of mechanism to look at translational projects with their own sort of rules.
It would also be nice to see on a percentage of the upside, so you could fund all of us.
That's nice.
Royalties to TMC. So,
Do one more question?
yeah,
No?
one more.
Are any results available for analysis when NIH funds the translational research?
I don't quite understand that.
In other words, the raw data, if things don't work out and maybe you want to resort it or resegment it, is that sort of more available to other researchers to be able to learn from when the NIH is funding the translational research and corporations?
I don't think so. I think NIH doesn't want to publicize their bad news either. So no, I don't think so. I mean, you have to basically just look at what's published. That's all the information you're going to have. You're going to have to look. There were negative trials published. So.
One of the themes today was efficiency. Could you comment on whether you think it might add to overall drug discovery efficiency?
Right. That's a very good question. Efficiency is, I would say, a factor of 10 greater through this program than what a pharmaceutical company will do. Easily, a factor of 10. You could do 10 times more work through this kind of mechanism. You know, the amount of money that we're spending is maybe not at the I&D phase, but if you add the entire thing together, all the infrastructure, all the overhead, and all the other things that a large company has, it's easily a factor of 10. So yes, it's much more efficient. If the NIH did this with the right people and the right programs and more of it, I think it would be a fantastic return on investment. And that's sort of like the whole concept of the CTI that Pfizer's doing is and it's an experiment right now. They knew if you gave $60 million to Scripps, it's going to be the same outcome. But if you give it and you shepherd it better, I think the efficiencies actually will be, maybe they're not tenfold, which I think they are in our case, but they could easily be 3 or 5-fold. But again, you need to have positive examples before you're willing to invest more in that.
Why is it, though? Why is it more efficient? And what aspects make it more efficient?
Why is it more efficient? Well, it's more efficient. Well, obviously, salaries at academics aren't the same as you don't have 15 VPs that are waiting to go to the meeting to tell you about the result, and then they go off and tell the other VPs. So I think there's a lot of more employees in large companies and relatively little research. You know, again, the budgets are skewed towards the other aspects of big pharma. Whereas we do 100% research, right? Or 95% at Tufts Medical Center, I guess.
For every one of you, there's at least 9 others that aren't having that. So.
Well, I think there's many successful people in this room. So yes, I think that's self-selected for also. Yeah.
In terms of efficiency, with the negative data, with our 3 screening, all the NIH's initiative to screen 300,000 to 50,000 compounds, all of that is dumped into a public database immediately after they screen. So in terms of being able to see negative results, that stuff is
It's also true for gene therapy studies. A lot of NIH-funded gene therapy studies which aren't published are actually available on databases so that other investigators can look at source adverse events which otherwise would not be reported widely to the community.
So I'm sorry to interrupt this. Perhaps we can pick this conversation up in another venue, but we should reconvene in the auditorium. So filter back in about 5 minutes. Thank you, Athan and Lidija. Thank you so much.